as we've already discussed in class, both the sympathetic and parasympathetic divisions are on in your body the whole time. They both at any given moment have some tone. That means influence on your involuntary effectors. But the rate of activity for each system can change from moment to moment based on your body's needs. But there is still this background activity, and the background activity can allow for an increase above that activity or a decrease in activity by a single system. Some involuntary effectors have more tone from one division than the other. For example, sympathetic tone causes about 50% vasoconstriction throughout your body of blood vessels. Increasing or decreasing sympathetic tone can in turn change your vessel diameter. When the sympathetic postganglionic neuron fires onto the smooth muscle of the tunica media of a blood vessel, and it fires more than its background amount, this will increase vasoconstriction. If it decreases its background activity, decreases how much epinephrine is being released or norepinephrine, then blood vessels will vasodilate. This is definitely something I'm going to test you on. How increasing sympathetic tone leads to more vasoconstriction and decreasing sympathetic output can lead to more vasodilation. When it comes to the GI tract, there's more parasympathetic tone, meaning the parasympathetic division has more control of the GI tract than the sympathetic. If we have more parasympathetic response of the GI tract, we're going to see more gut motility and release of digestive enzymes. If we block parasympathetic output to the GI tract, we will see less gut motility and less digestive enzyme release. We might even expect our patient to become constipated. I've asked you to know the effects of special targets of the autonomic nervous system. In class, I asked you to know specifically the eye, the heart, the lungs, and the gut, and how they reply, or respond, I should say, when there is an increase or decrease in sympathetic and parasympathetic tone. In class already, I showed you that the eye has both sympathetic and parasympathetic targets. When we're specifically talking about the pupil, as shown here in this picture, I showed to you in class that the iris of the eye has two distinct areas of smooth muscle. There are some smooth muscles that go in a sphincter or circular-like fashion, and then there are some smooth muscles that are more radial, like the spokes of a bike. It turns out that the radial fibers have alpha-1 receptors, one of the receptors for the sympathetic division. When alpha-1 receptors are stimulated, we get pupillary dilation. The parasympathetic division innervates the sphincter muscles, the circular ones. Those have muscarinic acetylcholine receptors. When the muscarinic acetylcholine receptor is stimulated, we would see pupillary constriction. We would also see accommodation or focusing of the lens for up-close reading. We can get pupillary dilation in another way, can't we? In addition to stimulating alpha-1 receptors, we could also block muscarinic acetylcholine receptors. What is the drug I've taught you that blocks muscarinic acetylcholine receptors? Did you say atropine? You're correct. So again, like our water faucet analogy, if we want more hot water, don't we have two options? Increase the amount of hot water output and decrease the amount of cold water output. If you block parasympathetic division, then expect to give more control, more tone towards the sympathetic side. And then you have to ask yourself, what would I see in my patient if the sympathetic division has more control for a target effector? Of course, there are many glands in your body. Are we talking lacrimal glands, salivary glands, GI glands, or sweat glands? Remember, the parasympathetic division has more tone in your digestive tract, but also over your lacrimal gland. If we stimulate muscarinic muscarinic acetylcholine receptors on these glands expect to see more parasympathetic output, meaning more secretions. The exception, of course, is the sweat glands found in the wall of the body, the dermis. Remember, sweat glands are involuntary effectors, and when you were first developing, this had a parasympathetic anatomy, meaning your pre-ganglionic fiber released acetylcholine, and the post-ganglionic fiber released acetylcholine onto the sweat glands. The sweat glands have the muscarinic acetylcholine receptor. But the brain centers took over this control, and now these involuntary effectors in the dermis of your body, the wall of your body, are under the influence of sympathetic control centers in your brain. I just talked about the GI tract. Again, mostly parasympathetic influence for the overall activity of smooth muscle and secretions of enzymes. If you need decreased gut motility, we need more sympathetic influence, and these will target through alpha-1 receptors closure of sphincter muscles, so you have less gut motility of food, chyme, and fecal material through the gut. When we think of a sympathetic response of the heart, we think of the pounding heart. There is an increase in both rate and force of contraction. The fancy word for rate is chronotropy, and the fancy word for how forceful the contraction of the heart is called inotropy. Do not confuse inotropy with ionotropic receptors that we've already talked about during this semester. Of course, the parasympathetic division will decrease heart rate. It has little to do with the force of contractility of the heart, and I will explain that in Unit 3. Blood vessels. Most blood vessels are under sympathetic tone. If we reduce sympathetic output, we get more vasodilation. If we increase sympathetic output, we're going to get more vasoconstriction. Beta-2 receptors are going to be a unique exception in this conversation, and I talk about that in the next video. With respect to erectile tissue found in the penis and even in the clitoris, this is caused by parasympathetic output causing erection through vasodilation of the erectile tissue, like the corpora cavernosa. This is caused mostly not from acetylcholine release, but rather from nitric oxide, which we talked about in a previous lecture. And we talked about Viagra and the Brazilian wandering spider. Just to review again, and we talked about this in class already, the parasympathetic division is considered to be craniosacral. By craniosacral, we mean that's where we find the cell bodies for the preganglionic neuronal cell. Cranio refers to nuclei in the brainstem that will give rise to parasympathetic branches for cranial nerves 3, 7, 9, and 10. Cranial nerve 3 is called the oculomotor nerve. The preganglionic fibers will synapse with postganglionic cells that are within the facial region, and the postganglionic cells are going to synapse with the smooth muscle found in the iris of the eye, the sphincter muscles, and the ciliary body that has smooth muscle as well that controls your lens and how flat or circular it is. And when it's when the lens is targeted just right, you can do up-close reading. This is called accommodation. Facial nerve is the name for cranial nerve 7. Its preganglionic fibers will also fuse, or synapse I should say, to postganglionic cells that are again found in the facial region. And the postganglionic cells will target the lacrimal and submandibular gland and control your tear production and saliva production from the submandibular gland. Cranial nerve 9 is the glossopharyngeal nerve, and the glossopharyngeal nerve will synapse to postganglionic cells near your parotid gland. And when the parotid gland is stimulated, you produce more saliva. Cranial nerve number 10. Cranial nerve number 10 is the vagus nerve. It will synapse to postganglionic cells near your visceral organs. And remember, these postganglionic cells are called terminal ganglia, and they are found within the wall of the effector. So vagus nerve is the only cranial nerve that extends into the thoracic cavity all the way into the abdominal cavity. It can extend to the left colic flexure, as shown in this picture. The left colic flexure is part of the large intestine. You have the ascending colon and then a sharp turn to the transverse colon. On the right side, this is called the right flexure, or the right hepatic flexure, right colic flexure. It has many names. And then the transverse turns into the descending, and that makes a sharp 90-degree turn as well. That's a flexure, a turn. So the vagus nerve can extend all the way down to the ascending colon and transverse colon, but after the left splenic flexure, or left colic flexure, then we need sacral equivalence. Remember, craniosacral. So that means for the descending colon, sigmoid colon, rectum, anything in the pelvic bowl like the bladder or even reproductive organs, we need S2, S3, S4. Those are the regions of your spinal cord where you have the sacrolateral gray column, and those preganglionic fibers will synapse to postganglionic cells in those involuntary effectors that are found in your pelvic bowl, like the descending colon, which is not in the pelvic bowl but leading to it, the sigmoid rectum, the rectum, bladder, and genitalia. As we've been discussing in class, you should be able to answer these questions. Again, your focus isn't so much on the anatomy, but in order to learn the physiology of these neurons and the pharmacology that goes along with it, you need an idea of their location. And as I tried to tell you, it's all about addresses. Where do you find them? So when is the parasympathetic active? All the time. When is it more active is the better question. Resting, digesting, peeing, and reading. Length of the preganglionic axon is very long. It gets all the way toward the involuntary effector, but it then synapses with its postganglionic neuron partner. So the postganglionic axon is very short. The location of cell bodies of the preganglionic nerve, I just told you this in the previous slide, craniosacral, nuclei for cranial nerves 3, 7, 9, 10, and sacral equivalents, the sacrolateral gray column of S2, S3, S4. Where do we find the postganglionic cell bodies? They're in the wall of the involuntary effectors. They're called terminal ganglia. What neurotransmitters are released by both of these neurons? For both, it's acetylcholine. When the preganglionic releases acetylcholine, the postganglionic cell body has nicotinic acetylcholine receptors. When the postganglionic neuron releases acetylcholine onto the involuntary effector, what receptor is used? Muscarinic acetylcholine receptor. The sympathetic division is called thoracolumbar. It's called that because that's where you find the preganglionic cell bodies, in a lateral gray column found between T1 through L2. Now that takes too long, so anatomists named that lateral gray column between T1 and L2, and it's called the IMLCC, or intermedial lateral cell column, which is a mouthful to say. When is the sympathetic division on or active? All the time. When is it more active? Fight, flight, which means to flee, freeze, and sex. Remember, we need the sympathetic division for ejaculation. The 4 F's, as I like to say. What's the length of the preganglionic fiber? Very short. If you look at this picture and you find the dashed black lines coming off the spinal cord, you can see how short the preganglionic axons are. If you look at the red lines, these represent the length of the postganglionic axon, which is much longer. Where do we find the location of cell bodies of the preganglionic neurons? They are in the IMLCC between T1 and L2. Where are the locations of cell bodies for the postganglionic sympathetic cell bodies? They are found in very distinct places. They are not found in the wall of the effector. They are either found in ganglia on both sides of your vertebral column, shown here with a straight vertical red line with little circles or ovals outlined with black. That's to represent the sympathetic ganglia of the sympathetic trunk. That is also called paravertebral ganglia. We also find postganglionic cell bodies on top of blood vessels. These are called prevertebral ganglia and also called collateral ganglia. And they also can have specific names for the blood vessel they're on, like the celiac ganglion, superior mesenteric ganglion, and inferior mesenteric ganglion. Your homework packet asks you to identify a fifth location, the adrenal medulla. The adrenal medulla itself is targeted by a preganglionic fiber. The picture here is erroneous. It's wrong. A preganglionic dashed line should go all the way to the adrenal medulla, and then the adrenal medulla should be colored red. The adrenal medulla is actually modified postganglionic neurons. The chromaffin cells there release epinephrine, but not as a neurotransmitter, as a hormone, into the bloodstream. Careful on this one, it will sneak up on you on the exam. What neurotransmitters are released? Well, typically, as we learned, the first one always releases acetylcholine, and the second postganglionic neuron in most places of the body for the sympathetic division will release norepinephrine/epinephrine. Norepinephrine is primarily used as a neurotransmitter, but not exclusively. Epinephrine can be considered a neurotransmitter too. Epinephrine is primarily used as a hormone, but not exclusively. The exception to the neurotransmitters released, of course, are in the body wall, as shown to the left of this picture. You see the piloerector muscle, the sweat gland, and blood vessels, and you're seeing a preganglionic fiber go all the way to them. Remember, this is actually going to have a parasympathetic anatomy. What the artists are not showing you is a very, very tiny, short postganglionic axon that would terminate on these involuntary effectors, piloerector, sweat glands, and tunica media of blood vessels in the dermis. Those postganglionic fibers would release acetylcholine. Again, that's the exception to the rule. Drugs that can affect cholinergic function. Remember, for both divisions, acetylcholine is released. For both divisions, the postganglionic neuron has nicotinic acetylcholine receptors, the one and only ionotropic receptor I asked you to know, where the receptor and the ion channel are the same entity. We've learned about muscarine, a toxin from the mushroom Amanita muscarina. Muscarine can stimulate all muscarinic acetylcholine receptors. The receptor primarily used on involuntary effectors targeted by the parasympathetic division, with the exception, of course, of the dermis and the involuntary effectors there being under control of sympathetic output. Pilocarpine is a drug that we use in a clinical setting. It too can activate muscarinic receptors. It causes profuse sweating and increased gut motility. Remember, the dermal involuntary effectors have muscarinic acetylcholine receptors, and that's why if we administer pilocarpine to a patient, not only will their heart rate drop and they'll have increased gut motility, but they will also have sweating. Nicotine can directly act on nicotinic acetylcholine receptors. Remember, both divisions use that on their postganglionic neuronal cell body. We also learned from our dendrites to synapse lecture about short-acting temporary cholinesterase inhibitors like neostigmine, pridostigmine, and physostigmine. I've also told you that we use another kind of stigmine called edrophonium This helps us determine if a patient is having a cholinergic versus a myasthenic crisis. When a patient who has myasthenia gravis, their immune system is attacking their nicotinic acetylcholine receptors, and the person has poor motor tone. Clearly, we would like them to be able to move. So we prescribe the short-acting stigmines. But if a patient overdoses on their stigmines, their muscles will fatigue, and by the time they present in a clinical setting, you will not be able to tell if the person overdosed or didn't take their meds in the first place. If you administer an extremely short-acting edrophonium, you can differentiate between the two. If you administer edrophonium and your patient improves, then that means they didn't take their medicine in the first place, and they are in a myasthenic crisis. If they don't improve, then you know they overinject or overdosed on their medications, and they're in a cholinergic crisis. We would then give that person atropine and protopam, and I explained why in our dendrites to synapse lecture. Cholinergic antagonists are drugs that block the muscarinic acetylcholine receptor. Atropine is the one that I've asked you to know. Here is a list of your sympathetic or adrenergic receptors. The ones highlighted in yellow are the ones I want you to know. Alpha 1, found on smooth muscle lining the gut, also found on the radial fibers found in the iris, and it's also found in smooth muscle of blood vessels. This causes closure of GI sphincters, more vasoconstriction, and pupillary dilation. I will explain when we're together in class how alpha 2 acts. It really acts through negative feedback inhibition to regulate the release of neurotransmitters, specifically norepinephrine. And I really do need to draw this out for you. Beta 1, we've talked about how many hearts do you have? One heart. So beta 1 is the primary receptor on the heart, not the only, but the primary. And this leads to increased heart rate, chronotropy is up, and force of contraction, that means increased inotropy. The story about the kidney to be continued. Beta 2 is the primary receptor found on smooth muscle found in your bronchioles. When stimulated, it causes bronchial dilation. Beta 2 can also be found on blood vessels. If beta 2 wins the battle and has more epinephrine bound to it, it will lead to vasodilation of blood vessels. And beta 3 is the receptor found mostly on adipose tissue. When it's stimulated, you have more utilage of your stored fats. You have lipolysis, and when you start to burn fats, you have more heat production. That's what I mean by thermogenesis. In the next video, I will continue on with drugs that can target the adrenergic receptors, drugs that can act as agonists, and then as the video continues on, you'll learn about antagonists that target the adrenergic receptors.